Right now, these nitrogenous bases line up along to make a long chain, and we call it a strand. This long chain is called a DNA strand. It is called a DNA strand. And typically, if we take a one cell, the DNA molecule has 6 billion nucleotide. So roughly around 6 billion nucleotide. So you can understand how long is the chain, because they line up at least one is 3 billion and the other side is 3 billion. So it's a huge long molecules with 3 billions of nucleotides. And we don't see amino acid maybe 1,000. Let's say 6,000, 1,820 amino acid. Glucose molecule, maybe 300 glucose molecule make the starch molecule. Cellulose, maybe 150, 100 beta glucose make the cellulose. But here, the DNA molecule it's a huge molecule because it has monomers repeating unit. The number is close to 3 billion. It's not a million, it's a billion, right? 10 to the power 9. So we can see the nitrogenous bases. This arrangement of nitrogenous bases store the genetic informations. It creates the codes. It creates the language on the DNA molecule. We will see this thing later on. So for example, we know how many nitrogenous bases: adenine, cytosine, thymine and guanine. So let's say example, this side has adenine, this side has cytosine, this side has guanine, this side has thymine. Now this is the one strand, and DNA molecule, to make a DNA molecule it needs 2 strands. So let's say another strand, then we call it a DNA molecule. 2 strands make the DNA molecule. Now if we try to add the other side, the DNA molecule, it is turning opposite direction. So it's like this. Same way, this should be the phosphate, and here we can find the nitrogenous base. But here you see it's a 3 prime end is here now. Does anyone know why it is turning this direction? Why the second strand is turning to the other direction? So this end is now 5 prime. You see 3 prime is here, now this end is 5 prime. This is 5 prime, this is 3 prime. So this is going this direction, this is going this direction. Do you know why they have this different orientation? We call anti-parallel. Why they make an anti-parallel arrangement? Yes, I think if you can answer this question, probably your knowledge is quite high enough. There are some answers. Yeah, Osandi and Oneli, I think you are close. Yes, Disat. Yes. Yes, I think your answer is correct, because if they make this direction only, they can fit into the nitrogenous base, because the nitrogenous base can make the bond if they change the direction, and the one nitrogenous base can make a bond with other nitrogenous base because their molecular structure is fitting to each other. And after that, uh, in front of A, it should be thiamine. In front of C, it should be guanine. In front of G, it should be cytosine, and here adenine. Because they make a 2 hydrogen bond here, and here make 3 hydrogen bonds. And this base pairing, you can remember all straight line for A and T, and curve line C and G. So this is often referred to as Watson and Crick base pairing because the Watson and Crick they discover exact shape and the structure of the DNA molecule. Somewhere 1956 or 1952, they got the Nobel Prize by discovering the exact shape of this DNA molecule. But we should always remember it is not only only these two scientists contribution gave this information, but many scientists who did a lot of experiment before, their contribution counted, but finally everybody cannot share it, but they are a little bit quicker to publish the exact nature of the DNA molecule, and they give some information. So base pairing is, we call generally Watson and Crick base pairing rule. Okay, now once they make this the antiparallel strands, because of the one side it's very strong phosphate backbone, and the middle hydrogen bonds are a little bit of weaker, they start to arrange a different nature like this. It's called it acquires this double helix like this, so we call a double helix. So its shape change, and we can see DNA molecule like this as a double helix. So this is the structure of the DNA molecule, and you can see the two strands are not identical but complementary, because here A, C, G, T, the order is like this, and here T, G, C, A. So the order is different. But if I add more nucleotide, again the composition is also different from the nitrogenous base composition, different from one strand to the other strand. So they are not the same compositions. So they are not identical, but they are complementary to each other. All right, so the function of DNA molecule, we are actually coming to that in a big way in the future. One thing is they store all our genetic information that can pass from one generation to next generation, and also it has codes to make the proteins. We also are going to learn protein synthesis in the future. So it has codes. These codes can make all the proteins in the body, and by doing that they can control activity of the cells. So everything can be controlled by the DNA molecules. Right, if you can already copy this one, I can briefly give an idea how this DNA molecule can function in the in the nucleus. So it is like this. So if we take a DNA molecule, as I mentioned, it's a long molecule, and then they have different codes because they make a very long sentence using these four nucleotide A, C, G and T. So if we look at the DNA molecule different way like this, and we can find all along from one end to another end a nucleotide. This is like this. You will learn in the future when three nucleotides join together. If we read these three as together, it makes a code we call codon. And one codon give a code for one amino acid. So one codon give a code for one amino acid. So then all these provide code for different amino acid. And you know, when protein is making, these amino acids join together to make the protein. So now these protein structure codes are written on the DNA molecule. And then a region of this DNA molecule is called gene. And this gene responsible for one particular protein, to synthesize one particular proteins. Don't worry, we have to learn this, but I just mentioned, not that important at this level, but in the future we have to learn genetic codes, how these codes are read, and how proteins are synthesized in details. There's one MCQ next. Some of the things you haven't learned, but I think still you can find the answer for this. Yes, some of them correctly answered. Let's forget 1, 3, 4, 5 because you haven't learned mitosis yet. And you didn't learn mutations. It's coming up in the next lessons, future lessons. What is mutations? What is mitosis? And you haven't learned about the protein synthesis. And you haven't learned about the genetic codes and the genes. But today you learn the two strands of DNA molecule are not identical. So incorrect. Identical is incorrect. Two strands are not identical because one strand is like this, and if it is as adenine, thymine, cytosine, guanine, the opposite strand, which has again thymine, adenine, guanine and cytosine. Now if you take the number of nucleotide here, one order is different. One thing is their order is different, and the composition is also different, because this side adenine only 1, thymine is 3, cytosine 1, guanine is 1. If you take this strand, composition wise, it has 1 adenine and 3 adenine and 1 thymine and 1 cytosine, 1 guanine. The order different, the composition of nucleotide difference. Only the similar thing is phosphate group and the deoxyribose, they are same, but nitrogenous bases are different. The order is different and the composition is different, so they are not identical. The two strands of DNA molecule are not identical, so it's an incorrect answer is this. You will see in the next lessons, mitosis, DNA double before they're going to move into 2 cells. And mutation is a change of the sequence of DNA. And this one, genes are different proteins at the end. The sequence is different. And it determines the what exact protein synthesized by the cells. Right, we are almost finishing the biological molecules. I think once we finish the lesson today, you can submit the tutorial, the answers for the tutorial for the biological molecules. Right, now let's see what is RNA. RNA stands for ribose nucleic acid, and they are different in DNA because they have a ribose sugar, and they are different. They have a uracil, and again they are different. They have a single strand. They have a single strand. So these are the 3 major difference between DNA and RNA. Not like DNA, RNA again exists in 3 different forms. One is called messenger RNA. Second one is called tRNA, transfer RNA. Third one is called ribosomal RNA. Again, we will learn the function of each one in protein synthesis. But messenger RNA is a single strand like this. tRNA has a special shape like this, and it always carries an amino acid. Then the rRNA are folded to make the ribosome. So here, like this, and it's also combined with different proteins to make the ribosome. That's a RNA. Black one is a RNA strand, and these are proteins. This organelle is called a ribosome. So these 3 involved in protein synthesis. mRNA, tRNA, rRNA. We are going to learn this the future in details. So then the function of each one we can learn. Okay, so we are almost finishing biological molecules. Just to finish this one, there are other nucleotides existing in the cell other than DNA and RNA. So we have to look quickly what are the other similar type of nucleotide we can find inside the cells. Right, first molecule is adenosine triphosphate. So I can delete this one. ATP. If you look at the ATP molecule, this has a ribose sugar and it has a nitrogenous base adenine, and it has a 3-phosphate group 1, 2, 3, and especially this between the phosphate group a lot of energy is trapped. Right, this ATP molecule can hydrolyze into adenosine diphosphate and a phosphate group is released. So ADP is adenosine diphosphate and the third phosphate is released. So by doing that, they can release energy. And this energy is the universal energy for all organisms. Bacteria to large animals, they use ATP as the fuel inside the cells. When we take glucose as energy source, when we take carbohydrates as energy source, these glucose molecules break down. So I hope you can remember this reaction. Carbon dioxide, water, and energy. So we write the aerobic respiration take place in the cells. Glucose break down to carbon dioxide and water and energy. So then glucose break down the energy released used to form this molecule ATP. Here the large amount of energy is going out to make the ATP molecule. So we are learning in the next section the unit 2 aerobic respiration, how glucose molecule provide energy to make ATP molecules. So ATP molecule is the universal energy molecule for all organisms. So this is again hydrolysis. So breaking the bonds is hydrolysis, and making this is called phosphorylation. That is adding a phosphate group to ADP molecule is phosphorylation. So if we can break ATP molecule, we can produce the almost 30.5 kilojoule per mole, which is a good amount of energy we can take from ATP. So structurally they are resemble to nucleotide as it has ribose, sugar, nitrogenous base, and instead of 1 phosphate, they have a 3 phosphate group. So do you have any questions? Hydrolysis of ATP can yield energy. So this is the producing of ATP by adding energy to ATP molecule from glucose. Glucose energy is transferred to ATP molecule. Okay, even though you haven't learned all these things, I think still you can answer this question. So what is the right answer for this question? Yeah, I got some of the answer. Let's see what is wrong with these answers. It has a pentose sugar, yes, and adenine and phosphate groups. Phosphate groups, right? It doesn't say how many phosphate groups, it just says phosphate groups. But look at this word. It's called nucleoside. So nucleoside is the pentose sugar with nitrogenous base. The nucleoside is a structure which has sugar and the nitrogenous base. So it does not have phosphate group. We are going to learn in digestion of nucleic acid. So there we learn nucleoside and all these things. Because when we take animal or plant cell inside, there are a lot of nucleic acid. So this nucleic acid digests into smaller compound during the process of digestion. So they are breaking into small pieces. So one of the intermediate is nucleoside. Can be produced by oxidative phosphorylation using solar energy. Okay, this sentence, oxidative phosphorylation, you haven't learned, right? Actually, this word, you're going to learn photosynthesis. The exact process is called photophosphorylation. The right word is photophosphorylation. It's very tricky that this oxidative phosphorylation and photophosphorylation is very close. We can easily trick to the answer too. But I think if we sharp with this number, hydrolysis to ADP releasing 30 kilojoule per mole is the correct answer. Hydrolysis can yield 30 kilojoule. I think there's a small spelling mistake here. It should be hydrolysis. if I remember, directly taken from this question from the exam paper. So they have a small printing mistake in the exam paper. Is formed pyridine. Okay, you haven't learned this one, but today you learn it has a ribose sugar. ATP has a ribose sugar. Okay, we're going to learn again the rest of the nucleotide, but just if you can remember the name, it's enough. There's another nucleotide, nicotinamide adenine dinucleotide, and flavin adenine dinucleotide. We're going to learn in respiration these two molecules in detail, and in the photosynthesis we're going to learn NADPH, nicotinamide adenine dinucleotide phosphate. So these three molecules mostly they act as a cofactors. So I mentioned cofactors. Some enzyme need additional molecule for its function. So if the enzyme is like this, and binding of one of these molecules to the enzyme activate the enzyme. So these are called cofactors. So these are organic cofactors, so they can activate the enzymes. We will learning in respiration and photosynthesis where exactly these molecules are involved and how they produced during the reactions. Okay, we did the biological molecules except the practical parts. Food test, reducing test for reducing sugars and non-reducing sugars, Benedict test, and iodine test and biuret test and a lipid emulsification test. They are not that difficult, small test, except the biuret test, Benedict test. It has a few steps. I will show these practicals next time. It doesn't take long. So next time, if you can print the cell workbook, we can start to learn the cell. Then after that, enzymes, photosynthesis, and respiration. So we can learn those things, right? Do you have any questions about today's lesson? At the end, there is MCQ questions. These questions I set as a MCQ tutorial online form. So you can send these answers for the question to me by clicking the right answer and submit at the end. So you can find the link from the website at the end, submit online tutorial, then you click it, you can submit the tutorial. You may need some knowledge for the Benedict test. For those, some of the questions, at least one or two questions, information about the Benedict test may require, but others you can write answers. Right, if you have questions you can ask. What are pyrimidine and purine? Yes, pyrimidine is just the word. And the other one is purine. So this is two groups under pyrimidine and purine, we can place the nitrogenous bases can be divided into pyrimidine and purine. Purines are adenine and guanine because they have two rings. And pyrimidine are cytosine, thymine and uracil, and they have one ring. It is just a grouping nitrogenous base based on the structure of the nitrogenous base into purine and pyrimidine. So any other questions? Okay, if you don't have any other questions, go through the notes and be confident with the theoretical knowledge, because you need very clear theoretical knowledge to answer the questions. So I am waiting to check how many answers you can get correct for the tutorial. So you can send it to me before the next class, because I can check, evaluate individually your levels, and I can track individually where you are, right? Okay, so I'm going to stop the class today, and see you again next time to start the new lesson, the cell, right? Okay, take care and see you later.